It's an honor and a pleasure to be here to celebrate with you all at this exciting event. And I am a relative newcomer to this exciting field of the intersection of neuroscience and AI, but I'll try to show you today why I find it so promising. And maybe I'll say as a sidebar, I tried to avoid it for a bunch of years. It was sort of happening around me in my department, and I thought, oh my God, do I have to learn all this fancy machine learning stuff? I'll just keep my head down and wait for it to blow over. And it didn't. And eventually I started looking around and saying, it's pretty damn cool, so I'm going to try that too. So my starting point for this work is the great progress I think we've made in the field of human cognitive neuroscience over the last 25 years. And that work has revealed this kind of new picture of the human brain. And what this diagram is meant to show is that there are lots of regions of the cortex in humans for which we have a pretty good idea what that little patch of cortex does. And often it's very distinctive and highly functionally specific. For example, a patch of cortex involved in recognizing faces, listening to music, understanding language. And each of these regions here is present in pretty much every normal person. We could pop any of you in the scanner and in a couple hours find all of these regions in your brain. And I think this is cool because I think of it as an initial, very rough sketch of the organization of the human mind. On the other hand, this is an extremely rudimentary picture that leaves the most fundamental questions unaddressed. For example, what actually are the computations that go on in each of those little patches of cortex? How might this systematic organization arise over evolution and development? And why is this a good way to design a brain in the first place? And I used to think that these were kind of the cool questions you toss off at the end of a talk, and I didn't really have any sense that we'd be able to answer them, at least in my lifetime. But I now think that we can actually chip away at them in a pretty serious way using artificial neural networks. So what I want to do today is give you just a taste of some of the efforts we've made in my lab to try to use these AI methods to try to understand what's going on in those regions of the brain. But what I want to do first is just give you at least a glimpse of some of the kind of evidence for that picture. It's my usual talk is a whole talk on that. I'm not going to do that. I'm just going to give you a taste to give you a reason to believe at least part of this. So I'm going to do that with the case of the fusiform face area, a little patch on the bottom of the right hemisphere, roughly in there, in me. And that region has been shown in dozens, maybe hundreds of functional MRI studies to respond much more when people look at faces than when they look at anything else. And that's nice, but functional MRI has its limitations. And actually, there are many other methods that show the same thing. And I want to show you some, I think, even stronger methods from intracranial studies in humans, in the rare cases where we get to study a patient who has electrodes in their head for clinical reasons. So this guy was a patient in a neurosurgery clinic in Japan a few years ago, and here are the locations of the electrodes all over the bottom of his brain. And to show you why I was excited to see about the locations of those electrodes, here's the bottom of my brain, showing you in red my face-specific regions, in green, a little hard to see, my place-specific regions, and the purple bits are the bits that respond more to color than grayscale. So the neurosurgeons told me that this gentleman was willing to look at our images while we recorded neural activity from those electrodes. And indeed, when we look at the responses of the electrodes in this region here, which is running along his right fusiform gyrus, along the bottom of the back of his head, of his brain, you see electrodes, excuse me, like these two here, where this is time in this axis, my cursor showing, yeah. And so red is the response to faces over this roughly 2 seconds here, and all the other colors are the responses to the other stimuli. So you see those little patches of his brain respond extremely selectively to faces compared to other kinds of visual stimuli. And that's nice, but in neuroscience, we want to not just measure responses to stimuli, we want to test the causal role of those regions of the brain in perception. And so the neurosurgeons had decided for clinical reasons to electrically stimulate those same electrodes. And I'm going to show you a movie of what happens when this guy is stimulated in those electrodes. And he's speaking Japanese, but there's subtitles.
そ 全部。また違うさ。違うふうにどんな感じ？頑張ってないんですけど。ね。まだあれですね。もう一回。
He's a perfect subject. Wants to tell us exactly what he's experiencing. He has no idea there's a face-specific region of the brain. He has no idea what part of his brain is being stimulated. He's just asked to tell us if anything changes.
なんかアニメできそうな感じのキャラクターの顔になって変わったんです。はい。口はそんなに変わってなかったんですけど。
That shows the causal role of that region in face perception. Here we wanted to know how specific is that causal role? Does it also affect your percept of something that isn't a face?
最初に1秒ぐらい、始まって1秒ぐらいがなんか、横になんか目、口って感じでちょっと一瞬見えたんですけど、これ何かなと思ってたら、気がついたらもうこの箱にしか見えて、なんすか、またさっきの横に、
This is a Japanese character on a card here. Okay, that's causal evidence, right? It's rare. We don't usually get evidence that clear, but in those rare cases where we get a person to volunteer to tell us what they experience when they're stimulated in a little patch of the brain, it gives us this extremely valuable and I think very striking evidence for the very specific causal role of that region in face perception. I won't take the time to show you now, but when he was stimulated right next door in these color preferring regions, he saw rainbows on top of whatever he was looking at. I know it sounds like I made it up, but I didn't. So there's similar evidence for most of these other regions here. I won't go through it. That's just to give you a gist of the kind of evidence behind this sort of a claim. So how can we use artificial neural networks to go at these questions here? Let's take this first one. What computations might be going on in each of those regions? It's all well and good to say that patch of cortex is really, really specifically involved in face perception, but that's not a very precise claim. How do we do better? Well, until recently, we just didn't have computational models that could do this in anything like the way humans can. And that's why it was big news to people like me when convolutional neural networks were suddenly achieving near human accuracy at face and object recognition. Because for the first time, we had at least one working model of what might be going on in the brain. And so the real question is that in fact how brains work, anything like that? And so there's a lot of ways to test that question. But first, let me point out it's really not obvious a priori. In fact, there's many reasons why it seems implausible that what's going on in a convolutional neural network would have anything to do with what's going on in the brain. First of all, these networks are not designed to match brains at all. They're extremely different from brains in hardware, most obviously, in learning rules, in training data, etc. So how do we test whether in fact they're similar to brains? Well, there's lots of methods, but maybe the most standard one is to build what are called encoding models based on these CNNs. And so just very briefly, what you can do is measure the response of a neuron or a voxel in the brain or a whole region of the brain to a bunch of natural images. Okay, so this is data from one of the first papers to do this by Dan Yemens and Jim DiCarlo. So here's the response of a single neuron in IT cortex, high-level visual cortex in a monkey, Here's its response to lots of different kinds of stimuli. These are all faces. So this is like the data I showed you in humans. So then what you do is you take these same natural images and you feed them into your network. And you collect a bunch of activations from the network, just as you can collect activations from the brain. Then what you do is you try to fit a linear model based on your ANN activations. It's basically you just come up with you fit a weight to each of these units in whatever layer you're using from the model, such that when you take the activations times the weights and sum them up, you get the response of that neuron. And you do that on a subset of the stimuli, and then you fix the weights and you ask whether you have in fact gotten a predictive model by whether it works for the held out stimuli. And so that's the question. Here's Yaman's and DiCarlo's data. In red is the predicted response to those same stimuli from that neuron using all the other stimuli except for that one in each case. So these are the kind of data that have been taken to argue that, yeah, this is evidence for some kind of similarity. I'll get back to in a moment what kind of similarity between these convolutional neural networks and brains. And I think that's cool for many reasons, but one of my favorite is I think it's some of the most beautiful evidence you could ask for a claim that David Marr made a very long time ago, when he said, the nature of the computations that underlie perception depends more on the computational problems that have to be solved than on the particular hardware in which their solution is implemented. So in other words, it's the task that's the problem that's being solved, the task that constrains how the solution works. And this is beautiful evidence for that, I think. Okay, so then I had nothing to do with this stuff I've talked about so far. But then my postdoc Ratan Murthy said, hey, we can do that with our functional MRI data. Let's try building artificial neural network-based models of some of our favorite whole big regions of the brain, like the face, place, and body selective regions in the ventral visual pathway. And at first I was like, huh, what? How would that go? Why would you do that? What would that tell us? I don't know. It's really his idea. And he did it, and it worked magnificently. So he scanned subjects looking at 185 different images, trained up ANN-based models in the way I described before. And here each dot is a stimulus, the predicted response of the ANN model to that stimulus, and the observed response on held out data. And you can see the correlation between predicted to observed is 0.9, not corrected for the noise ceiling, just 0.9. And that means these are spectacularly accurate predictive models of the responses of those regions of the brain to new stimuli. And so that's cool, but what do we do with this? Well, actually lots of things. There's lots of times when you just can't run a functional MRI experiment on thousands of different stimuli, for example. So one of the first things we did was say, well, Kanwisher has been claiming for 20 some years that face area responds more to faces than anything else, but she's only tested a few hundred stimuli. How do we know? How do we know which other ones to test? Well, so Ratan ran his predictive model on the entire machine learning image database to see if he could find any images that were predicted to produce a very strong response in the face area that weren't faces. And he and I manually checked the top 200,000. They were all faces. So it was a powerful possibility of falsifying my hypothesis, which was exciting. It didn't actually falsify it, at least yet. And then you can play all kinds of other games. We used GANs to generate predicted best images for each of these regions. We got something like that for the face area, like that for the place area, and that for the body selective region. So there's lots of cool things we can do with these models, and that's fun. But I want to mention a bunch of questions and limitations of these kinds of models. First one is, if these things are hypotheses about what might be going on in the brain, how well can we choose between those hypotheses? Because there's lots of different kinds of ANNs, different architectures, different training data, different all this stuff, and we want to be able to choose between them. And the problem is this method doesn't choose between them very well. Lots and lots of different kinds of models that are very different all fit the data pretty well. So here's some of our data showing a bunch of CNNs trained supervised, unsupervised, a bunch of vision transformers. They all have really good predictivity for voxels in the ventral visual pathway, better than untrained networks, but they're all really similar to each other. So that's a problem if we want to use this method to choose the best model. A second problem that's bugged me all along is, are these encoding models kind of too low a bar for establishing similarity between CNNs and brains? And so, you have all of these units, hundreds or thousands of units in some layer, and you get to pick your own weights, and like, it's maybe not that surprising that you can fit some complicated function when you have so much to choose from. And does that really mean that when it works, that network is doing anything similar to brains? I don't know, but it has bugged me for a long time. So my postdoc Meenakshi Khosla, who just started a faculty position at UC San Diego, and who's fantastic if any of you are applying to grad school, she came up with a solution to both of these problems. And that is, instead of building these linear weighted combination predictive models, let's get really literal. Let's do one-to-one matching, a single unit in a layer of a network to a single neuron or voxel in the brain. And let's see if that works. And actually it works really well. And so I think that's really interesting, both because it's a much more literal match between networks and brains. It makes me believe them a little bit more, that network really is a lot like a brain. And second of all, it better enables us to discriminate between models. So with linear predictivity, you can't discriminate the fit of CNNs from vision transformers. But with this one-to-one matching, the CNNs fit better than the vision transformers. So we think that's promising. A third question about these models is, we need them to fit not just neural responses in the brain, but we need them to fit behavior, human behavior or monkey behavior. So do they? Well, let's start with face perception. With Katarina Dobbs, who was my postdoc a few years ago and now has her own position at the University of Gießen in Germany, we decided to look at the huge literature on behavioral signatures of face perception in humans. And people like me who've been studying face perception for a long time collected these behavioral signatures, and were very pleased with them. Here's our precious little signatures. We didn't ask why the face system behaved like that. We just collected them and say, see, faces are special. But that's kind of a theoretical thing to do. We should also ask why the face system behaves like that. And Katarina her work, I think, gives us some nice answers to that. So what are these signatures? Well, there's lots of them. One is just that we're really, really good at face discrimination. Another is that we humans have a pretty characteristic face space. That is, if you ask people how similar pairs of faces are, they have a characteristic spacing of different faces that seems to describe some kind of representational space in the head. A third one is the face inversion effect. We are much worse at recognizing faces presented upside down than right side up. And that inversion cost is greater for faces than for any other stimulus class that has been tested. And another one is the other race effect. Whoever they are, they all look alike. If you haven't spent as much time around them, that other face type, you are less good at discriminating them. It doesn't mean you're racist, it just means your perceptual system has not been tuned up with the diagnostic features of that other race of faces. So we wanted to know, why does the human face system exhibit these signatures, and whether CNN models could capture these behavioral phenomena, and which models. So the hypothesis we were testing is that these distinctive behavioral signatures in the human brain are simply the result of optimization for face recognition. And if that's true, then we might expect that CNNs optimized for face recognition would exhibit all the same phenomena, but CNNs optimized for something else might not. And so Katarina found a lot of evidence for that. CNNs trained up on ImageNet do not fit the face data very well. CNNs trained on the VGG face database or other face databases fit these data beautifully. And further, it's not enough to just expose the network to all the same faces. You need to have a loss function in which the network has to discriminate one face from another. Okay, so we took that as evidence for this hypothesis. And so in terms of what computations, I showed you that ANN models can predict neural responses in category selective regions of the human brain and behavioral signatures of face perception. And I think that at least suggests that the computations underlying human vision are at least somewhat similar in some way to those in CNNs. Though, as I'm sure you guys know, there are lots of notorious things that ANNs still don't capture in human human perception, from adversarial images to 3D shape perception and on, and there's lots of work to be done in those domains. But I want to go on and consider this question of how might this organization arise in the brain? And so in addition to the face, place, and body regions I've talked about, there are two other, well, there's a bunch of them, but I'll talk about two other selective responses that you can see. Over here is the visual word form area that responds selectively to orthographies you know. If you read English and Arabic, but not Hebrew, that region will respond to English and Arabic, but not Hebrew, and et cetera, et cetera. It has to be trained up by your individual experience. And recently, around a year ago or so, Meenakshi and I reported that a data-driven analysis of responses in the ventral pathway revealed what appears to be a food selective response, that is a selective neural response to images of food. Since then, we have some new data that doesn't exactly fit with that hypothesis, but I'll leave it here for now and just say it's quite food selective. And so if you think about how might that stuff arise in human brains, well, the first thing you'd think of is like these categories really matter to us humans, right? They're like the perceptual front end of a lot of stuff we care about and that feeds into cognitive operations we carry out on these stimuli. So for example, face and body perception can be seen as the front end of social cognition. Who is that person? What are they doing? Place perception as a front end for navigation. Text perception feeds right into reading and language. And food perception, I don't know, staying alive, pretty important. So that's a natural hypothesis. But there's an alternative, and that is maybe the meaning and significance of these categories just isn't necessary for them to arise in that part of the brain. Maybe just the statistics of visual input would be sufficient for the brain to develop these specialized regions. So that's very hard to test in humans, but we can test it with CNNs by asking, because CNNs don't know anything about the significance of these categories to humans, we can ask whether they too develop similar selectivities. And so Katerina has done some work in that vein, and so she took CNN architectures and trained them on a mixture of faces and objects, 1,000 different faces, 1,000 different object categories, and just concatenated all the labels here and mixed in all the stimuli during training. The network wasn't told these are faces, these are objects, they were just all mixed together. So she trained up the network. And the first thing is that network, of course, knows nothing about the difference between faces and objects. It's just trained on that input. It did really well at both face discrimination and object classification. And then we looked inside to see how it was organized. And so the way Katerina did that was to use a causal lesioning method on the network. So she looked at one of the late layers in the network, and she sorted the units in that layer by their causal role in face perception, and she lesioned out, zeroed out the top 20% most important units for face perception, and showed, of course, a huge drop in face perception, but preserved object classification performance. And then when she did the opposite, knocking out the top 20% most important units for object perception, of course, object recognition performance dropped precipitously, but face recognition performance was preserved. So this shows that that network had spontaneously segregated itself into separate pathways or separate sets of units for face and object recognition. So in some sense, there's some kind of covert segregation in the network that it spontaneously evolved, just like we see in the brain. And I think that's cool because it says that that this network didn't need to be given any strong priors about faces or the distinction of faces, and it figured out on its own that it should allocate separate units to face perception. So what about other categories? So Meenakshi's been taking on this question with a slightly different method. So she used data-driven analyses of functional MRI data to find major functional components in the functional response in the ventral visual pathway, and also in CNNs trained self-supervised on ImageNet. So what she did was to feed in both the brain and the CNNs the same set of 1,000 natural images into the brain or into the networks, and to collect activations. So for both the brain and the network, you get this big matrix of 1,000 images by a whole bunch of CNN units or brain voxels. And then you basically factorize this matrix. The details don't matter. She used Bayesian non-negative matrix factorization. It gives you these components, which include a weight distribution over the brain or over the network units, but also importantly, functional response profiles of inferred functional components. So the question is whether the functional components that arose in the brain and the networks would be similar, even though the networks don't know what these categories mean to humans. So that's the question. And so here's what she finds in brains. This was the paper that led to the report of food selectivity, but it also, even though it was completely data-driven, it discovered as its top 5 components with the matrix factorization method, selectivity for faces, places, text, and bodies, 4 categories we already knew had selective responses in the brain, so those were nice positive controls, but it also found this food selective response. These are the 1,000 images color-coded by the salience of place information, face, food, text information. The color is the salience of that category in that image. Okay, so that's the brain data. Now the question is, when you do the same analysis on networks, feed them the same 1,000 images, do the matrix factorization on the networks, what do you see? And you see something pretty similar. You see NMF components that have selective responses to places, faces, food, text, and bodies, quite similar to what we saw in the brain. And so I think what this says is that since similar selectivities are arising in ANNs that know nothing about the meaning of these categories, that suggests that brains may not need very strong category-specific priors to build those category structures in their brain. Doesn't mean they don't have them, it says they might not need them, right? And that seems to be true across a wide range of architectures and learning rules. Okay, so that's cool, but how much of the brain might be captured by current artificial neural network models? Well, I've just described, some of the work from my lab, but there's lots of related work going on in other labs. My colleague Josh McDermott down the hall from me is doing beautiful, beautiful work on the auditory system, showing that ANN models trained on natural auditory stimuli can also strongly predict responses in auditory cortex, including with the layer-wise correspondence where more primary regions in purple are better accounted for by the earlier layers in the network and higher level regions by later layers in the network. And of course, lots of people, not including me, are using recurrent neural networks to model neural signals for movement and for navigation. And my colleague Ev Fedorenko and lots of other labs are now using large language models to model behavioral and neural responses to language in the brain. Here's Ev Fedorenko's data showing really nice neural predictivity in language cortex using functional MRI data, really well modeled by GPT-2. And so I think that's really cool and empowering. But how far is this going to go? I mean, probably not everything. I doubt that the current kind of ANN models that are available are going to be able to model all of these other bits of cortex. I think that's a really fascinating open question. It may be that some of the mental functions that go on in our brain that have more to do with building models of the world than just kind of recognizing patterns may require very different kinds of architectures and different kinds of networks. And I think that in itself will be informative. And maybe, just maybe, that will eventually enable us neuroscientists to something that will provide clues for AI. And I'll stop there. I don't want to burn up too much time. A question?
Yes, so we will have plenty of time later on, but we said that maybe one quick question, if somebody has a burning question you want to ask, feel free.
I was obviously extremely lucid, not a single question.
Well, yes, we have one question of you.
So the CNN models, they heavily rely on the distribution of the training data. So how do you make sure that your training data for your model is the same as the data that a human would see on average?
Yeah, it's a great question. If you didn't hear it, how do we know that the CNN models have the same training data as humans? We don't. In fact, we know they don't, right? But a lot of people are now collecting the data we really need by sticking little video cameras on infants' heads and collecting the actual developmental input that infants get. And then it'll be super interesting to see if those models fit even better.
Thank you very much. So please join me in thanking Nancy once again.